Hi, everybody. It's Doug Beavis, and today is our first ever Health and Nutrition AOCS e-pitch by students. So very excited to have you here. We have four great presenters today. And we have a panel of judges. I'm the current AOCS president, so very nice to meet you all. Thank you for coming. And our judges today are Dr. Fabiola Dionisi. She's a senior expert and lipid network leader at Nestlé in Switzerland. Dr. Holiday Durham-Zanetti, she's a clinical investigator from Nutrilite Health Institute, Amway. And Dr. Matt Miller, research scientist from Cawthron Institute in New Zealand. So what's going to happen today, each student will present for up to about 5 minutes, including a PowerPoint. This won't be the ePoster. And we're going to watch the chat area, I guess, for important updates. And after each student presents, a 5-minute question and feedback session will happen. You can post any questions you want to in the chat area, and we'll try to address as many as we can, but we're limited to about 5 minutes. So, and then when all these students are done presenting, we'll have an open audience poll where you actually vote. So that's very important because we want you to vote for your favorite pitch, and that counts towards the winner. So first place prize is $200, and second place is $100. So I've seen the presentations, a lot of really good work, very talented people. I was a student in 1994, and I know my research wasn't nearly as rigorous as this. So we're going to begin with our first presenter. It's Julie Etienne. She's from France, Inserm, and she is an engineer in bioinformatics. She graduated from INSA in Lyon, France. She's currently a PhD student in the CarMeN, Cardiometabolic Diabetes and Nutrition Laboratory in France. So, and the title of her talk is Uptake of Fatty Acids by Enterocyte, New Insights Gained from Mathematical Modeling. And Julie's co-authors are Armeli Penhote, Maria Carolyn Mikulski, and Kara Nibble. So we'd like to introduce Julie to present her 5-minute presentation.
Hello, everyone. I'm very happy to be here and to present my ePoster at this AOCS meeting. My ePoster deals with the fat contained in our meals. And this fat is mostly composed of triglycerides, which carry fatty acids. In more details, my ePoster focuses on how these dietary fatty acids are being taken up by cells of our intestine, namely enterocyte. In the literature, this process of uptake is very debated, and this process of uptake is also usually reviewed as being composed of passive diffusion along with a carrier-mediated mechanism. We decided to question again this uptake of fatty acid by the enterocyte by asking ourselves if the observed uptake curves were compatible with the pathways suggested by the literature. In order to answer this question, we use mathematical modeling and data fitting at the interface between computer science, biology, and mathematics. And we wrote 3 models of uptake. So first, model M0 considers an uptake only by passive diffusion. Then model M1 considers an uptake both by passive diffusion and a carrier-mediated mechanism. So this is the model suggested by the literature, model M1, and moreover, the carrier E is not clearly identified yet in the literature nowadays. And finally, model M2 considers an uptake by passive diffusion and this uptake is being followed by an enzymatic transformation of fatty acid inside the cells. And this we know occurs because fatty acids are being transformed to be reesterified into triglycerides. So what did we, to make this a bit clearer, we could imagine fatty acids as being baby sea turtles getting out of their eggs, and we wonder how they cross the beach in order to reach the sea and find other sea turtles. So do they cross the beach alone? This will be model M0. Or do baby sea turtles cross the beach alone, but some are using some kind of carrier like a seagull, like in the movie A Turtle Sail? Or do babies cross the beach alone, but they are somehow encouraged by adults waiting for them at the end of the beach, and once they reach the sea, they feel part of the group, and it's somehow like motivating. So now what did we do with those three models? We studied them and we compared them to experimental data extracted from research papers. So as a reminder, I recall the three models on the left-hand side of my presentation. And now let's have a look at our results. So looking at the graphic on the top, we can see model M0, where uptake is only by passive diffusion. And we can see that this model hardly reaches the experimental data or stays in the lower part of it. And moreover, if we play around with the parameters, we see that volumes have more impact than permeability. Then in the middle, we can see model M1 and M2, and we can see that both models equally fit the experimental data using nonlinear regression. And finally, at the bottom, the graphic shows the maximal uptake of our three models, and model M2 has a greater uptake potential than model M1, with, as a reminder, model M1 being the model suggested by the literature. So as a conclusion, what can we say about the uptake of fatty acids by the enterocytes? Well, first, the pathway suggested by the literature, so here model M1, is compatible with the experimental data. So this is actually reassuring, even if it does not answer the debate. And another interesting point is that other pathways are also compatible with the data. For example, here model M2. That suggests, here model M2 suggests that maybe it is more efficient for the cell to invest in enzymatic transformation of fatty acid inside a cell than into membrane carrier. And last but not least, if our mathematical modeling provides new insight into this uptake of fatty acid by the enterocyte, it also highlights the need of new experimental data in order to quantify and differentiate different uptake fluxes, if more than one. And we hope this step is also the first step of the idea of modeling a full virtual enterocyte. Thank you for your attention, and I am ready for questions.
Hey, thanks, Julie. That was a great presentation. I really like the comparison. Absorption of fatty acids is incredibly important, and there's a lot of malabsorption problems in our world. So it's really a neat model that you have. So right now we're going to talk about questions, I guess, from the judges for you. And we've got our three judges, and they're going to ask some questions.
So I can start. Thank you, Julie. Great work. Well done. Well presented too. I would like that you elaborate on the limitation of this type of modeling.
Well, I guess first is all the hypotheses that we make to model, because it's a model, it's not the reality, and it's hard to know what really happens inside a human or living organism. So I guess this is one of the limitations, maybe the biggest, I guess.
And in terms of error, what type of error is associated to your points, let's say?
Well, first is sorry.
The variation. What is the variability of your curves?
Well, we did not look in the variability. It's more how for example, when we fit the models to the experimental data, how well it fits to the data. So it's the error of the fitting. But in terms of variability, we have, for example, the variability on the parameters we fixed from the literature, or the ones we inferred from the modeling. And for example, in the literature, there are many values of parameters, and we just chose one, but we didn't take into account the standard deviation, for example. So this will be an improvement for our modeling.
Okay, thanks a lot.
Thank you.
Hello, hello, Julie. Great presentation. You said you sort of ground truth this against the literature. Have you got any plans to test this in vivo and in vitro, and how would you go about that?
This is a good question, because in my lab, we work also on CACO2 cells, so cell mimicking the enterocyte. And the idea would be to be able to differentiate which fatty acids are being taken up by passive diffusion and which are being taken up by transporters, if any. And I guess this is a wide question, because in the literature it was quite a lot studied before, like in 10 years ago maybe. And I guess it's hard to get out every genes that could be involved in this uptake, because we don't really know how many and which, and then we don't, I don't know if we know how to cut off the passive diffusion, for example. This would be an interesting idea. Or I know they also talk about endocytosis. So this is another mechanism that maybe involves proteins. So I guess there is many lines of research for experimental experiments.
Thanks, Julie.
Nice job, Julie. Thanks for your presentation. So are there plans in your lab to follow up any of these bioinformatic work that you've done, or?
Yes, it will be great to have new data points, for example, to have to improve our modeling. Okay. It's still a topic of discussion inside the team.
Okay. I like the fact that you've used mathematical modeling. It's something very different in this field to assess this. So thanks for that contribution.
Thank you.
Okay. Yeah, thank you. I think putting a model on something really allows you to look at lots of different experimental variables and start to define things. So, neat research. Thank you very much, Julie. So we're going to head on to our next presenter now.
Okay, thank you very much.
Welcome back. Hey, our next speaker is Snigdha Guha, and she is a graduate research assistant at the University of Nebraska in Lincoln. Snigdha grew up in New Delhi, was educated there, did her bachelor's and a master's degree there, and she's currently working on the role of dietary bioactive peptides in modulating metabolic disease. This includes looking at bean-derived bioactive peptides' efficacy to reduce vascular inflammation and related pathophysiology. So the title of Snigdha's talk is Efficacy of Dietary Gamma-Glutamyl Peptide in Modulating Vascular Inflammation. And her co-author is Kostov Mahumir, who's the assistant professor at Nebraska. So welcome very much, and we look forward to hearing your talk.
Thank you. Hello, everyone. Today I'm going to pitch for my research, which is titled Efficacy of Dietary Gamma-Glutamyl Peptide in Modulating Vascular Inflammation. So let me start my pitch by asking this one question. What is the #1 cause of death in the US? And the answer is cardiovascular diseases, or CVDs. In fact, it is the #1 cause of death globally. And one fun fact here is that it has been found that if a person follow all of these 4, that is, doesn't smoke, controls their BMI, eat lots of fruits and vegetables, and exercise on a regular basis, they can actually reduce the risk of CVD by 85%. But sadly, only 3% of Americans have been reported to follow all these 4. And according to WHO, it has been reported that 1 in every 4 deaths in the US is because of cardiovascular disease. The picture here on the right is a summary which signifies the importance of inflammation in causing heart disease, because low-grade chronic inflammation is the main initiator of endothelial dysfunction or plaque formations in the blood vessel, which actually leads to disorders like atherosclerosis or hypertension, which are the major risk factors for cardiovascular disease. Now, the problem with the current pharmacological interventions for CVDs are that they have various side effects, as well as in severe cases, patients have to undergo vascular surgeries, which are very expensive. Therefore, in order to control such a growing epidemic, we need more of a preventative approach. And for that, my research proposes use of food-derived bioactive peptides. And one class of those peptides are gamma-glutamyl peptides. So these gamma-glutamyl peptides could be naturally found in foods such as beans, cheese, onion. And it has been seen that when gamma-glutamyl peptides interact with the calcium-sensing receptors, or CASAR, which are on our tongues, they give rise to a kukumi flavor. And this same interaction with the CASAR has also shown to have anti-inflammatory effects, which was previously proven in gastrointestinal inflammation. And since calcium-sensing receptors are ubiquitously present in many mammalian tissues, including vasculature, therefore my research hypothesizes that gamma-glutamyl peptides can reduce vascular inflammation as well in CASAR-dependent pathways. So this is a summary of my research method and results. So for my research, I chose gamma-glutamyl valine, or gamma-EV, as my model gamma peptide. And I cultured human aortic endothelial cells, which are blood vessel cells from the aorta. And I first pre-treated them with gamma-EV for 2 hours at different doses, like 0.01, 0.11 millimolar. And then I induced inflammation using tumor necrosis factor alpha for another 6 hours. So when inflammation is induced, there is increase in expression of inflammatory biomarkers like selectins, cellular adhesion molecules, chemokine, and cytokines like IL-6, IL-8. But it was found that in the presence of 1 millimolar gamma EV dose, the expression of these biomarkers was significantly reduced, suggesting that gamma EV had anti-inflammatory role. Then we also found that this anti-inflammatory effect was calcium sensing receptor dependent, because when we used a CASAR specific antagonist, which was NPS 2143, which specifically blocks CASAR, the anti-inflammatory effect which we previously saw was abolished. We also looked at some key signaling molecules which might be involved in this mechanism, which included beta-arrestin, CASAR itself, then P65, which is a molecule of the NF-kappa B pathway, as well as Cre binding protein. The next part of my research focused on seeing whether this gamma peptide could actually cross the intestinal cells, because it is important to see if we consume them, they should be able to pass intact and reach the blood vessels. So for that, we grew intestinal epithelial cells CACO2 on permeable inserts and added gamma EV on the apical side, and then allowed them to transport for 2 hours, after which we looked their or checked their concentration in the basolateral layer. And we found that gamma EV did pass intact through the cells. We also looked at the mechanism of transport of gamma EV, and it was found to be both paracellular, which was between the cells through tight junctions, as well as through peptide-1, which is through a peptide transporter. Therefore, the relevance or the long-term goal of this research is, with such promising results, we could actually use gamma peptides to develop nutraceuticals or use it as a functional food ingredient, which would be effective in reducing vascular inflammation, and therefore might provide a novel strategy for prevention of cardiovascular diseases. Thank you so much for your attention, and I can take some questions now.
Well, thanks, Snigdha. That was a great presentation, and what a very interesting topic. Again, an example how food can have significant impacts on physiology in our lives. And, makes me want to eat more beans. So, but again, we'll have the judges ask you questions, and we'll go from there.
So this is Holiday. Nice job presenting, and I like the fact that you're using plant-based interventions. So that's awesome. I just had a question about your results. What did you find? Tell us more about kind of some of the conclusions that were made from the work that you did.
Okay, so we started with a single peptide, which is γ-glutamyl EV or γ-glutamyl valine, which was previously reported to show anti-inflammatory effect in gastrointestinal inflammation. So we wanted to see whether this peptide could actually also cause reduction in vascular inflammation. And so we did see that there was a dose-dependent effect for this peptide to reduce inflammation in endothelial cells, and it was effectively able to transport across the intestinal cells. So moving forward from here, since this is just one peptide, and as my research focuses on bean-derived peptides, second part of my research also involves with extracting these peptides naturally from beans and seeing their efficacy. So there would be like a mixture of γ-glutamyl peptides, which we have already extracted from beans. And eventually, and we have seen that they can reduce inflammation in vascular cells. So they might be used later on for as an ingredient for functional food, which something like that, like supplements which you can take, and then it would be helpful in prevention of these cardiovascular diseases.
Thank you. Out of curiosity, what types of beans?
We work with Great Northern beans. That's the most produced variety in Nebraska.
Okay, good to know. Thank you.
Yep.
Thanks a lot for your presentation. Very interesting. In the bean, these peptides, they exist as a peptides or as a part of a protein?
So yes, these peptides, they actually exist as peptides. So they are part of non-protein nitrogen sources. So you can actually extract them as it is. So when we extracted them, we did not digest them or anything. They were just present naturally as peptides.
Okay. And during the digestion, so let's say that you ingest these peptides, do you think that they would reach the gut?
Actually, we have not checked that yet, but in literature, it is suggested that because these peptides are gamma-glutamyl peptides, and because of their unique gamma bond, which is unlike the usual alpha, which is like the alpha carbon, which forms the peptide bond, they form from the gamma carbon of their of the glutamic acid. So because of that unusual bond, they're actually resistant to gastric digestion. So they can survive the digestion which is happening in our gut and can actually cross the intestine and reach the blood vessel. So that way it is pretty unique as well to be used as an ingredient.
Thank you very much.
Okay, and a couple of questions from me. You mentioned earlier that they produce a kokumi flavor. Could you describe what that flavor is?
So yes, it's called the sixth flavor. So when you eat food like miso soup, if you know, they have that what do you call umami taste. Do you know that?
Yep.
So yeah, so basically it was actually first discovered in miso soup. So these peptides were found to be the major constituents of the soup, and then there was research which identified these peptides and also linked them to their interaction with the calcium sensing receptors on our tongue, which was the main reason for these for the perception of this taste. And then later on, the studies showed their anti-inflammatory effects as well.
And following on from that, so you say cheese, beans, and onions are good sources. What is the best source? If you've got to name a source, what bean or cheese or?
For example, I have been working with Great Northern beans, but we have also looked at some other beans such as garbanzo beans, and I found that the gamma peptides were a little lesser than that. So I think in order to give a complete answer, we need to test all beans because their concentration would vary among beans. I have just worked with 2 beans, and I can say Great Northern beans really have a good amount of gamma-glutamyl peptides, which is not so much in, for example, garbanzo beans. So, and yeah, but I haven't like looked at all the sources. So,
Great, thank you.
Okay, how much? What kind of concentration are we looking at? Like micrograms per gram?
Actually, in beans, it's nanograms. So it is, to give you a number, it's like 53 nanograms per gram. I know the concentration is very low. So few of the limitations is, one of the limitation is actually that. And then to overcome that, we might even look at some processing, like for example, fermentation of the beans has been shown to increase these gamma peptides in literature, which is another side of the project, which is going on currently in our lab, to see if by any processing methods, we can actually increase the content of gamma peptides. But this is just like a fundamental step or like a starting point just to see if these peptides are biologically active or not. But then once we find that, there will be like other processing methods to actually increase their concentration, maybe breeding plants in a way to have these more. So yeah, but currently it's pretty low.
It's just curious. It's fascinating to me. I know I've seen some studies with fish dipeptides that have big effects on vascular responsiveness. So thanks very much. That was a great presentation. And,
thank you.
Appreciate it. So, I believe we'll move on to our next speaker. We'll get All right, we're back. Our third presenter today is Koki Tsujimoto. He is from the Kansai University in Osaka, Japan. Koki, thank you for being with us today. I know it's late in the evening in Japan, so we appreciate your efforts to be here. So Koki has degrees from Kansai University, and he's currently in the graduate program there of science and engineering. And he is a doctoral student. He's studying changes in lipid metabolism through the lipid intake. And they've successfully prepared scallop oil from internal organs of scallops, one of my favorite foods. And the title of the talk today is Effects of Dietary Phospholipids Prepared from the Japanese Giant Scallop on Cholesterol Metabolism in C57 mice. So, Koki, take it away.
Thank you, Mr. Chairperson, for introducing me. Hello, everyone. My name is Koki Tsujimoto from Japan. I'm pleased to the opportunity to tell you about some of our recent work on effects of dietary phospholipids prepared from the Japanese giant scallop on cholesterol metabolism in C57 black 6G mice. The current time in Japan is 3 o'clock a.m. This is the time when I normally be sleeping, but I'll do my best to win the hour. First, I explain about the scallop internal organs. In Japan, the quantities of 450,000 tons per year of scallop are landed, but 40,000 tons per year of scallop internal organs other than adductor muscle are discharged. The internal organs contain N3 polyunsaturated fatty acids and phospholipids, but they also contain toxic substances, so it couldn't be used effectively. We have successfully prepared scallop oil called as SCO, containing low toxic substances from the internal organ of scallop. SCO has high EPA and phospholipids. Therefore, we aim to produce SCO as a health food material that can be a source of N3 puffer. Our previous studies have confirmed to be non-toxic of SCO. In addition, we have demonstrated dietary SCO decreases the serum and liver cholesterol contents in mice, and this effect couldn't be found in other N3 puffer containing oils, such as menhaden oil and krill oil. In the present study, we investigated what are bioactive substances in SCO that lower serum and liver cholesterol content using mice. At first, we have separated the phospholipid of SCO called as SCOPL. As you can see this table, 4 oils were used for animal tests. Soy TG is soybean oil, soy PL is soybean lecithin, SCOTG is almost the same fatty acid composition as SCOPL, and SCOPL is SCO's phospholipid fraction. 5-week-old male C57 black 6J mice were fed high-fat and cholesterol diet. Experimental diet contains 3% soy TG, soy PL, SCOTG, and SCOPL. After 4 weeks, mice were sacrificed, and we analyzed serum and liver cholesterol contents, fecal lipids, liver and ileum messenger RNA expression levels, and liver CYP7A1 protein expression level. Next, I explain the results. Dietary SCOPL diet significantly lowers the serum and liver cholesterol content compared with soy TG diet. Since this effect was not observed in the SCOTG group, the serum and liver cholesterol content lowering effect of SCO intake is considered to be the effect of the phospholipid fraction. It was partly mediated by 2 points. One is the enhancement of the liver CYP7A1 expression level. CYP7A1 is the rate-limiting enzyme in the classical bile acid biosynthesis pathway. The other is enhancement of fecal neutral sterol excretion. Soy PL had no effect of increasing liver CYP7A1 expression level. Therefore, we focused on the increasing CYP7A1 expression level, which is characteristic of SCOPL group, and investigate the mechanism of increased liver CYP7A1 expression level. Next, I will now explain the summary of results. This slide shows the mechanism of the enhancement of liver CYP7A1 expression level in the SCOPL group. First, the results show increased expression of CYP2C70, which is involved in uricolic acid synthesis in the liver, and increased beta uricolic acid content. Second, taurine conjugated beta uricolic acid is secreted from the liver into bile and absorbed to the ileum. Beta uricolic acid is antagonist of FXR and then reduces FGF15 expression level. FGF15 is known to regulate CYP7A1 expression level. FGF15 is transported to the liver and suppresses the CYP7A1 expression level. As you can see in this slide, SCOPL is thought to increase liver CYP7A1 expression levels through this mechanism. These results show that SCOPL lowers cholesterol content through an increase in CYP7A1 expression level, which was not seen in fish oil or soybean lecithin. Therefore, SCOPL can be a health-promoting component with cholesterol content lowering effect. Thank you for your attention.
Thanks, Koki. Great presentation. Enjoyed it very much. Very interesting, the effects of different phospholipids and fatty acids on cholesterol metabolism. So we're going to open up questions now to the judges, and we'll go from there.
I might start off this time. 3 o'clock in the morning. It's 6 o'clock in the morning here in New Zealand, so I know how you feel, Koki. It's very early. I just want to ask a couple of questions. The amount of polar lipid in the scallop, what sort of percentage polar lipid compared to triglycerides are there in your scallop oil?
Thank you for your question. About 20% of SCO is phospholipid.
Yep, and you did your studies with your comparison to soybean oil. Would you think that a fish oil might be a nice comparison? In fact, that they have a similar long chain omega-3 fatty acid profile where soybean hasn't, and that might elucidate the differences between the fatty acid profile or the delivery platform, whether in the lipid class.
You can mean phospholipid class,
or just like the triglyceride form of just a fish oil. Compare the scallop versus a fish oil to see whether the fatty acid profile, or if it's something uniquely in scallop oil that is actually having a benefit.
Sorry, I didn't fully catch your question. Could you speak a little slower?
Yep, no problem. Have you considered comparing your scallop oil against a fish oil rather than a soybean oil?
Sorry, what? Fatty acid composition, fish oil compared to scallop oil?
No, in your in vitro rat or mouse model, have you considered looking at a fish oil, like a different control rather than a soybean?
Okay. SCO have high N3 polyunsaturated fatty acids, for example, EPA and DHA. But soybean lecithin have no N3 puffer. Fish oil have N3 puffer, but fish oil have no phospholipid. So SCO characteristic have SCO have characteristic have N3 puffer and phospholipid.
Okay. Yep. Thank you.
Sorry. Thank you.
Thank you very much for your presentation. I would like to understand, in case you would like really to give, you know, this component to human being, how would you tackle this? Would you separate and purify these phospholipids, or are you planning to give this liquid, this extraction directly to human beings? So which format actually?
Sorry, one more please.
So in other words, if you wanted to implement your solution, how would you do? What is next step? What is the next step for your research? What are you going to planning to do next afterwards in your laboratory?
My future research?
Yeah.
Yes. I want to find bioactive substances in SCOPL in future research. SCO's phospholipid fraction is component of about 10% ceramide amino acid phosphate. It is a characteristic sphingolipid with a carbon phospholipid carbon phosphorus bond. I think this is a bioactive substance with this effect. So I evaluate the effect of ceramide amino acid phosphate on cholesterol metabolism in the future research.
Okay, thank you very much.
Thank you.
Any questions, Dr. Zanetti?
I just wanted to follow up kind of with Fabiola's question. How would this be applied to human models? The research that you've done in mice, but how would you apply it to a human model?
It have been reported N3 puffer is used for human. So SCO is also SCO is effective for human too.
Okay. Okay. Thank you.
Thank you.
Well, thank you, Koki. Appreciate your presentation. Very well done. And look forward to hearing more about scallop oil in the future. So,
Thank you.
We'll move on to our next speaker. And today we're going to have our fourth and final speaker, Gustavo Reyes Pulido. So thank you for joining us. Gustavo is a PhD student in the Tecnológico de Monterrey, Mexico City. Gustavo earned his bachelor's degree from the Tecnológico de Monterrey in Mexico City. He moved to London to pursue his master's degree in biopharmaceuticals at King College London. And the title of Gustavo's talk today is Triborheological Analysis of Reconstituted Gastrointestinal Porcine Mucus under Different pH Conditions. Welcome, Gustavo.
Hello, hi everyone. Thank you for joining this presentation. Actually, as Doug mentioned, I will talk about the triborheological analysis of reconstituted gastrointestinal porcine mucus under different pH conditions. Actually, it's important to mention first that mucus is a biological barrier that covers every wet epithelial surface in our body, so it's quite important. As well, the mucus is a natural hydrogel that is formed by the entanglement of mucins. Mucin is an important protein because actually it gives the mucus their viscoelastic, their structural, and some of their functional properties. So it's important to study it. As well, I want to mention that the mucus protect us from viruses, bacteria, external particles, but also it is a barrier for the drug delivery or some molecules that we want to deliver. So the objective in this research is to characterize in a better way the mucus, actually using tribo and rheology in order to understand more about these mechanical properties of mucus and also how the pH in the gastrointestinal tract change the mucus properties, because I want in the future to talk about more drug delivery. The results that I have obtained in terms of rheology, we can see in figure 3, there is a graph in which I plot the modulus, the elastic and the viscous modulus of samples. I performed the experiment under 4 different pH. There are the more common pH in the gastrointestinal tract, the pH 2, that it's very acidic in the stomach, pH 4 is the transitional pH, and pH 6 and 7 that are more related with the intestines. As we can see with the black dots that are related with the acidic conditions, there are anelastic dominant properties. It means that under the stomach, mucus acts more like a gel. It's a protective gel because the harsh acidic conditions could start to auto-digesting our stomach. So that's why it's more like a gel. But in the neutral pH, probably 6, 7, with the blue and green figures, we can see that the modulus are quite similar. Even there are very slightly crossovers, which means that the sample is more, it performed more like a viscous polymeric solution. So it's more like lubricant properties than protective properties. As well, in figure 5, we can see that independently of the pH, the viscosity of all the samples is reduced when the shear rate is increasing. It means that mucus acts as a lubricant, even if it's on a gel or it's on a solution. Of course, on a solution is a better lubricant, but at the end is lubricant under all conditions. Finally, I perform a tribological analysis through a Strybeck curve in order to know how the coefficient of friction interacts with the mucus. As it was expected, the pH 2 or the acidic condition exhibit a higher coefficient of friction because it acts more like a gel. The function of mucus here is more to protect. But on pH 6 and 7, the mucus exhibit the lowest coefficient of friction because it's more lubricant. That's why it's a very good thing to understand these kind of conditions. The relevance and the future perspective of this project, first of all, the relevance is because studying the viscosity and the coefficient of friction under condition that mimics the human body conditions, it's important to understand more about this functional barrier because it's quite important to know all the properties. Because in the future, as I mentioned, the perspective is to develop some nanoparticles for drug delivery. And one important interaction between nanoparticles and this mucus is the mucoadhesivity, which can be evaluated through triboroology, but also can tell us if the nanoparticles are modifying the structure or the properties that should be very important, for example, the protective and the lubricant. So that's why I am focused now on characterizing mucus in order to pass in the future for the interaction with these nanoparticles. Thank you for your attention, and I want to acknowledge Medina's group, Tecnológico en Monterrey, CONACYT, the Mexican Council of Science and Technology, and also the AOCS annual meeting and expo. Thank you.
Thank you, Gustavo. A wonderful presentation. Something I didn't realize about mucus. So, it certainly is a very important barrier and conduit to transmission of things into our body and keeping bad things out. So, great talk. Judges, we have some questions for Gustavo.
Yes, so thanks a lot, Gustavo. Very interesting topic. Can you please elaborate more about implication and a possible application also of your research?
Well, thank you for the question. Actually, it's quite interesting because the application of this research, first of all, is to know more about the properties that has this biological barrier in terms of the protection and the lubrication that are one of the main characteristics, because mucus has an isoelectric point around 2.6, 2.7. So that's why it changed their structure from a gel to a solution. That's the first and important thing to mention here. And also because, as I mentioned, in the future, I want to start to performing some experiments with different polymeric nanoparticles, because there is a recent interaction that has been studied that is called mucoadhesion or bioadhesion with mucus, in which the nanoparticles could interact through different mechanisms with the mucus, but this interaction sometimes could affect this change, this structural change. And it's important to keep these changes because, well, as the mucus is a protection. So if, for example, in the stomach, it acts more like a solution, probably it could affect our digestion because the acids could auto-digest the stomach. It's a very extremely example, but it's something to take care. So that's why I am focused on this. I want to know first all the properties and then how to interact with nanoparticles.
Thank you very much.
Thanks, Gustav. As with Doug, I didn't know much about mucus, so quick question around, is all mucus the same? So you showed in your rheology that it changes with pH, but is the compositional or the chemical properties of mucus the same in your stomach as it is in your lower intestine?
Thank you for the question. Actually, it's a very interesting question because, well, actually we have mucus in almost every part of our body. For example, also in our nose, in our mouth, in our lungs, among other places. But the important thing is that each organ has different composition in the mucus. In general, it has like the same elements, but the concentrations are quite different. For example, in the stomach, the main component is mucin. That is the most important protein here. The concentration there is around 5-6%, but in our intestines is a little bit less. It could be around 3-4%. Assuming that the person is healthy, because also during diseases, this concentration can be modified. So also it's important to mention that because as well as the concentration is different, so the behavior is different. Also, it's there are differences in terms of the thickness of the mucus. For example, there are places where we have around 100 nanometers of thickness, like in the small intestine, or in the rectum, for example, there could be even up to 800 nanometers thickness. So it's quite important to consider all these differences. So actually, in the future is one of the next steps. I want to consider all the different stage, well, the different concentrations and the thickness in order to see which area could be the best one for delivery and a specific kind of nanoparticle.
Cheers, Gustav.
Do you see opportunity for this in terms of lipid delivery systems, or kind of where is it in terms of what types of delivery systems you're thinking about in terms of the application of lipids? Or
okay, thank you for the question. Actually, lipids delivery systems are a very good ones because mucus is lipophilic, so it's a good one for the penetration. But in my case, I am more focused on polymeric delivery systems because, well, my idea is to have some delivery system that has more specific targeting, and I know more about how to modify the surface of nanoparticles, polymeric nanoparticles, in order to have a targeting ligands or different elements in order to specific targeting. But also lipids are a very good one options.
okay, have you taken also into consideration biofilms at all? Because I know that definitely plays into the availability of mucous membranes and things like that.
Sorry, can you repeat your question, please?
Have you considered looking at various biofilms and because that definitely interacts with kind of the mucus availability in different systems?
Okay. Well, actually, until now, I haven't considered the biofilms. I am more focused on, well, to develop or to study these interactions between nanoparticles and mucus with drug delivery idea. But also the biofilms is an interesting topic. I need to read more about it because I only have like the general idea, but it's an interesting topic.
Thank you. Nice job. Thank you.
No, thank you.
All right, Gustavo, thank you. Excellent presentation. Now I'm going to be thinking about mucus and potential impacts. So that wraps up our presentations for the e-pitch. Wow, just a big congratulations to all of you. Exceptional presentations, and I learned a lot. And I think we have a few questions from the audience. And I'll read. Okay, we have a question for Snigdha. Snigdha, can you pop your camera on? Hi, Snigdha. This question is from the audience. Do you think adding 1 millimolar of your peptide is high?
It is not high because we also looked at the toxicity on cells, and it was not toxic to the cells at all. And so we looked at a broad range, and we actually went up to 5 millimolar. And like after 5 millimolar, it was actually toxic to the cells, but 1 millimolar was not that high.
Thank you. And then a second question for you, Snigdha, was I think you addressed it a little bit. Can you scale up the production of gamma peptides with fermentation, like the example, making bread?
Yes. So that is some of the projects. So for example, we would try to do a bean paste or something with like more of a fermented product, which can be used. Yeah, so maybe like bread, but so that would also require like product development side to it. But for now, maybe like fermentation to produce some bean paste or something would be like is what I can see can be done, because fermentations using lactobacillus has been shown to increase gamma peptides. But yeah, definitely bread would be an option as well, I guess.
Okay. Excellent. And that's the extent of the questions from the audience. So now we have the audience voting. So the audience, you have to vote using the annual meeting website. If you're logging in from Facebook or YouTube, you can't vote for that through those things. So you'll have to go to the AOCS website to vote. So please vote for your top presentation. And this vote obviously is very important. It'll impact the placement of our top finishers. So the audience vote, as I mentioned before, counts as a single vote. So there's 3 judges and the audience's vote. So it adds up to 25% of the impact. So, and the audience poll will be open for 4 minutes. And so please vote for your favorite pitch. everybody. Results are in. So excellent presentations, great research. You're all on a really strong track to be great contributors and have a lot of fun in science. Our second place winner is Julie Etienne. So with her presentation, Uptake of Fatty Acids by the Enterocytes, New Insights Gained from Mathematical Modeling. So congratulations, Julie. $100 coming your way. So
thank you very much.
Great presentation. And the winner of today's competition is Snigdha Guha. So congratulations. Really interesting research. And
Thank
Izzy,
you.
clap, clap.
Thank you, judges and audience.
And Snigdha, you're going to get some $200. I'm not quite sure how it gets to you, but you can do something fun with that. So, and,
Thank you.
Yeah, so that wraps up our presentation for the day. Thank you all. We had quite a few viewers. I think we were in the 40s for our competition views, and this is, I believe, archived as well, so you can direct your friends and family to share your presentations. So excellent job, and thank you all for supporting AOCS, and I wish you all the best in your endeavors in the future.